Dear students, in this module, we're going to look at the primary structure of proteins. You know that protein structures are organized into 1', 2', 3', and 4' structures. In other terms, primary, secondary, tertiary, and quaternary structures. In this module, we're going to see only the primary structures or the 1' structures. How they are formed, what is the basis, what are the databases for these structures, what are the experimental methods to obtain these structures, and we'll overview all of them. So let's start. So you know that the 1' structures are essentially the amino acid sequences. So here in this figure, this is how the protein structures are organized. And currently we're looking at the simple amino acid chains as shown in this red box. The information available here is simply the amino acid sequence for each protein. So once the amino acids they polymerize, then they form these chains and they come together. As a result, charge, the polarity, the hydrophobicity, and other properties of the R groups within each amino acid are perturbed, and therefore this perturbation gives rise to a wider range of interactions for the entire polypeptide chain or the protein sequence as shown by this red line. Of course, later this polypeptide chain goes to form secondary structures, tertiary structures, and quaternary structures as well. Okay, so there are 20 different amino acids as you know, and each one of them is given here. What you need to understand from this slide is that there is a 1-letter code for each amino acid that is representative of this amino acid, and in case of a protein, we simply put these 1-letter codes together to form the entire protein sequence. So this is how a protein sequence forms, and therefore you can create information databases by simply listing the amino acids 1-letter codes that are there in a protein. There are two methods that are there to obtain the amino acid sequence of a protein. The first one is the Edman degradation. It's an experimental protocol that is slow but is able to tell you the amino acid sequence for up to 100 amino acids. The second experimental protocol is called the Tandem mass spectrometry, and this technique is able to sequence very large proteins as well. When I say sequence, I essentially mean the amino acid sequence. So once Edman degradation or tandem mass spectrometry outputs the amino acid sequences of proteins, they are collected in a database. There are multiple databases that exist. For instance, there is the UniProt, which is a very big protein sequence database. It contains a lot of other information as well, such as molecular weight, the PI, and other things. Also, there is SwissProt and many others. So the point is that once you sequence the amino acid chains, then this sequence information is stored in a database for further reference. So to conclude, the protein structures, they begin with the formation of 1' structures or the primary structures. The primary structures are essentially the amino acid sequences. An amino acid's 1-letter code is used to form these sequences. Edman degradation or tandem mass spectrometry helps you to obtain these sequences from the samples in experiments, and that once these sequences are obtained, you put them in a database for further utilization. Now, once you know that you have the 1' structure of the protein, this in effect is going to determine the 2' structure or the secondary structure of the protein as well.